Yes, thank you very much for the introduction and the opportunity to speak. I'm very excited about this opportunity because although, as Jeff mentioned in the introduction, I have responsibility for many of the industries, for many of the process industries up to the end of last calendar year. My main responsibility was pharma and food and beverage. And I have a lot of background myself in doing pharma projects. Jeff also mentioned mechanistic modeling, mechanistic understanding. I'll use these terms quite a lot during the talk. Sometimes though, we also use the term physical science-based digital twins. And this is to distinguish these approaches from approaches that are purely data-driven, data science-based approaches. And I will, in the beginning, explain why we focus on not purely data-driven, purely data analytics approaches, but that we try to use as much first principle knowledge as possible, and then we augment it with data. And we do this for two reasons. One is we need much less data. And when you're developing pharmaceutical processes, manufacturing processes, but even when you're operating pharmaceutical manufacturing processes, you often don't have lots of data. Yeah? So we can talk a lot about big data, but the reality is that very often we may have a lot of data, but it has very little information, or in many cases, we don't even have a lot of data. So the talk will be structured as follows. Let me just get the pointer. Should help a bit. So we're going to start off with a bit of an introduction. And during the introduction, I will talk about how models are used not only in commercial manufacture, but that the journey starts when we develop new processes, when we do the tech transfer, and ultimately we can use those models or digital twins, as people like to call them these days, in a commercial manufacturing setting. So after the introduction, which will take quite a bit of time, for about 15 minutes, we will go into 3 different case studies. Solvent swap, so this is synthetic pharma on the drug substance manufacturing side. The second case study is a case study that was presented by the FDA, which is around drug product manufacture and using models for developing a process, but also training operators and testing for robustness. This first case study actually is a case study that was presented 2 weeks ago at IFPAC by Janssen Pharmaceutica. The third case study is a biologics case study where we'll look at monoclonal antibodies and the ability to look at continuous manufacturing, but also batch or perfusion and so on. I'll then wrap up with some conclusions, and then there'll be at least 15 minutes for questions. Good. So without further ado, I'm going to start off with an introduction. Doesn't seem to be responding. Okay, here we go. So a number of trends that are important for this talk is that one of the drivers for all pharmaceutical companies to use mechanistic models, physical science-based models, is because there are fewer blockbusters, but if you want to generate the same revenue, you probably have to bring more products to market. Yeah? And you need to be able to do that with fewer R&D resources, fewer tech transfer resources. So this is one trend that is driving the uptake of mechanistic modeling approaches. Similarly, you can accelerate significantly because if you can do your DOEs, your design of experiments, on a computer instead of in the lab, you can save weeks, and in terms of continuous manufacturing, often months of development time. There is a lot of pressure from certain regulatory bodies, not all of them, but especially the FDA, to some extent EMA, and to look at more science-based approaches. And by that, I mean not purely data science, but actually physical sciences based on first principle understanding. And this, of course, one of the reasons why the FDA also actually uses the modeling techniques I talk about, and why they presented about some of the work there. The fourth trend is an important one as well, modernizing data and knowledge sharing. I think this is a general trend that regardless of whether you use mechanistic models or data-driven models, there is always a challenge in terms of curating data, having a single source of truth, making sure there is enough metadata so that you use the data in a correct and authorized manner. So some of the key implications of the trends I just mentioned is that if you continue to do things in a very data-driven approach, doing lots of experiments in the lab, lots of experiments at a pilot scale, maybe another pilot scale, then several trials at manufacturing scale, is that it takes too long and it's quite costly. So part of what I'm going to present today is we believe a key element of digital transformation of the way that pharma companies develop new drugs, develop the manufacturing processes for the drugs, do the scale up, the tech transfer, and ultimately, as you'll see in the, especially in the first case study, how you can also use these same models to operate much more efficiently and much more robustly. So the mission that we have within our organization is to help bring high quality drugs to patients faster, which I think everyone agrees with is a good thing to aim for. And we do this through combinations of providing software that certain companies use, as well as associated services. And what we specifically focus on is enabling people in the industry as well as academia. We probably should have mentioned at the beginning, I'm very actively involved in the PIPS, the Pharmaceutical Innovation Program, Singapore, where we also support many of the academic partners to be able to execute digitalization projects where process modeling plays a key role. So our specific focus as the part of Siemens I'm responsible for is that we enable people in industry and academia to quickly, rapidly create digital process twins. And the reason we can do this very quickly is because we don't need a lot of data collection. We usually only require data to calibrate a few parameters in the model. And in some cases, we don't need any data for calibration, but we use some data to validate the model. So this summarizes some of the key advantages and reasons why we focus on mechanistic understanding or physical-based sciences, and that we use data mainly for validating the model. So one of the advantages of models is that we can, of course, experiments, especially in biologics, can take days, weeks, but even in synthetic pharma, a crystallization experiment may take a day. And if you have to do a DOE with 2 factors, you can do it in a week. If you have to do 3 factors, you may be looking at a month or more of experimentation. So models, once we have the models, can be solved very efficiently. We can look at not 2 or 3 factors, but we can look at 10 factors on the computer to really understand which parameters are critical process parameters, which material attributes are really critical material attributes. So standing more quickly. The second point is the fact that because mechanistic understanding is based on first principle understanding of physics, chemistry, sometimes empirical understanding, but also understanding of biology, it means that we can extrapolate the knowledge, not infinitely, but within certain ranges we can extrapolate. And of course, with a pure database approach, it is very dangerous to do any extrapolation. And of course, when you are looking to do scale up, tech transfer, by definition, you are always looking to extrapolate. So this is another important aspect. Again, similar to the first point, if we run the experiments on the computer to look at 3, 4, 5, even 10 factors, we don't consume lots of API. We don't need lots of lab time. Again, we do need some data, but very targeted data to calibrate the models. So just to give you a bit of an idea, so when we talk about digital process twin, what is actually inside a digital process twin? And this is lots of different things. We have, of course, information on the equipment, which could just be volume, heat transfer area, the mass of the equipment for thermal inertia. If we have a catalytic process, of course, information on the catalyst, information on physical properties, whether it's solubility for crystallization or vapor-liquid equilibria, as we'll see for solvent swap, or it could be isotherms for drying operations. Information on control systems, how we operate the plant, what are the degrees of freedom, what are the constraints, and so on. This information all gets put into a model by people who don't do any coding. So this is, when I talk about modeling, it's not about writing equations, but it's configuring models with information on the materials and their properties, equipment, and so on. We put that into this kind of environment. This is a screenshot of the software which shows a bioreactor. We have some things like tangential flow filtration, chromatography steps, maybe an ion exchange chromatography, and so on. And once we have that model, we can, of course, use it to do simulations. And usually when people talk about modeling, they say modeling and simulation. We are of the opinion that simulations are, of course, have some use, but it's probably not the most useful thing you can do with a model, because simulations it's like trial and error. Yeah? You change some parameters, see what happens, change some parameters, see what happens. If you really want to get an understanding of the impact of all your process decisions, your disturbances, your material attributes, and so on process performance and quality attributes, in particular critical quality attributes, it is much more efficient to use automated techniques like global system analysis, which will calculate the uncertainty in the process as a function of input disturbances, variability. It will also tell you, if you have disturbances in 5 factors, which of the disturbances has the highest impact on the variability in your product. So this is an automated approach to look at essentially design spaces and not only to see how big or small your design space is, but how could you make it bigger? Because if you can make your design space bigger, your process will inherently be more robust. Now, our background, we started developing this kind of software in 2009, so 14 years ago. Initially, all the users in companies like Pfizer, GSK, Merck, and so on, Sanofi, Bayer, they used it mainly for process development and tech transfer, which is what we call digital design. In the last 2 years, we have seen more and more of these companies starting to use the models in commercial manufacturing. And you will see one example in the case study from Janssen, but there has also been work that has been presented by Pfizer and GSK and Eli Lilly on using this technology to support operations. Okay, so for us, it's very important to think about a model or a digital process twin as it's not something you use for one application, but you use it for many applications across the life cycle. And there are 2 reasons that we want to use the same model across the life cycle. One is that if we only have to develop one model and keep it up to date, it's much cheaper than developing a model for process developments and then developing another model to support commercial manufacturing. It also means that we can guarantee consistency in the knowledge that we use. And actually, a third advantage is that very often it's when people start using the models from development in manufacture, it helps break down some of the communication and cultural barriers that sometimes exist between the process development organizations and the supply chain commercial manufacturing organizations. Okay. Now, although we use, we enable pharmaceutical companies to allow models to be used across the life cycle, it's important to realize that people in process development who may have PhDs, will want to interact with these models in much more detail. They want to understand details of thermodynamics, kinetics, and so on. And these are important when the model originally gets developed. When we go to manufacture, most of the people in manufacturing are not interested in thermodynamics or kinetics. They are interested in sort of the performance of the process in terms of yields, purities, but also some of the safety constraints and operational constraints. So we may use the same model, but we have to be able to expose very different user interfaces. Sometimes the interface directly to a control system, sometimes it may be an interface to a display that the operator can use to decide whether to take any actions or not. And again, depending on how you set it up, the criticality of the model, from a regulatory perspective is very different, and it has other different implications for GMP aspects. So in brief, we have tools that are used by really engineers, chemical engineers, biochemical engineers for configuring, validating, and deploying models. And those deployed models then either go to web applications, which are used by people in manufacturing to say, well, actually, we have a different, the excipient quality has changed. We have a different specific surface area of our lubricant. Will we still be able to produce tablets with the right porosity, with the right tensile strength, and so on? And you can then use the model before you run the actual campaign to see whether the excipient is going to cause any problems or not. And then the third area is what we call digital applications, where we can take a model that was created by a biochemical engineer and put it behind a dashboard, link it up to maybe a DCS system or to edge devices, and so on. And then the models can be used for soft sensing. So being able to, many of the quality attributes that are of interest cannot be measured in line, or it's very expensive to measure them in line. So often impurities, of course, can only be measured in a lab. But if you're running a continuous manufacturing process, you want to have real-time information. If you're looking at things related to particle size distributions, again, typically they will be measured offline. If you have a model that runs in parallel with the real manufacturing process, you can predict impurities, you can predict particle size distributions, and so you can actually do real-time control and not waiting several hours before deciding whether you can continue or whether you need to actually reject a batch. So there are quite a few companies using this kind of advanced process modeling technology that I've been describing. Many companies, many industries, of course, several of the universities in Singapore, but also many of the academic institutions in Europe and US use this kind of advanced process modeling. And of course, the FDA, who have the second case study, are also using this kind of modeling technology. So, of course, if you work for one of these companies and you're interested in more details, you can always contact me, and we can put you into contact with the colleagues who are using the technology. Okay, so that's the introduction, and then I will continue to the case study. So the first case study is, as I said, is solvent swap. So this is drug substance manufacture. Then the second one will be a drug product manufacture, so small molecule and small molecule. The third one is large molecule or biologics. So this is a case study that was presented together with Janssen at IFPAC in New York a few weeks ago. What you see on this slide is actually the use of the model in the desktop environment I was talking about. So this is what the engineers in process development, process engineering will use to design an optimal recipe for a batch distillation, in this case, the solvent swap. What we will talk about in this presentation is how the same model is used to actually control this process in an automatic way and with flexibility to either run the process so that you minimize batch time, or you minimize solvent or energy consumption, or you actually have a cost function, economic cost function, which weighs the cost of materials, the cost of utility, but also the cost of occupying the asset and before it can be used for the next batch. So some of the challenges around solvent swap is it can be time and cost intensive. It's a manual, often manually operated. The final composition of the solvent API mixture is usually not measured in real time, which means you have to wait at the end before you can actually continue. Until you have to wait for the analysis from offline measurements. There are issues that can result in things like foaming or precipitation, which you would like to avoid. And with modeling, mechanistic models, you can use them to quickly, you can run the model many times to find optimal operating conditions for different objective functions. In this case, the model is derived purely from first principles knowledge of physics, chemistry, thermodynamics. So the model didn't require any data from the solvent system, from the API, to create a digital process twin. You can use the model to look at what-if scenarios. So if you have certain disturbances, or whether you can still actually run the process successfully to the end. And you can change the way you optimize. So if you have no problems with acid pressure on the acids, if you have more than enough spare capacity, then you may decide to purely look at minimizing the energy, the solvent costs. If you have a lot of pressure on the acids and you want to start the next batch as quickly as possible, you can optimize to reduce purely the batch duration. So this is a specific application for with THF and propanol as the solvents. The idea is, of course, to get the original solvent concentration has to be reduced below a minimum level. Whilst we optimize the process, the solvent volume and temperatures, of course, have to remain within acceptable ranges. And the objective, as I mentioned before, is to minimize batch time or energy or solvent costs, or a combination of those. And the technique that is used is that we take the model from the process development, and then in the manufacturing organization, we hook it up to a DCS, a distributed control system, and the nonlinear model predictive control uses the model to automatically calculate the set point trajectories for the supervisory control, for the flow rates, for temperatures, and so on. So we have in the online every few seconds, or maybe more frequently, and we can send information back to the DCS system. So before we get to the deployment in manufacture, we first of all configure and calibrate the model. Then we have to configure the optimization, set up the objective function. What economic objective function do you want to have for the business? And tune the NMPC, the not to into manufacturing. So if we encounter the model is a standard model in the library of the software. And it has mass balances, it has energy balances, it deals with heat transfer, heat losses, and of course, the most important thing is it calculates the vapor-liquid equilibrium within the drum. So I mentioned we didn't use any data to calibrate the model, because we don't need data for mass or energy balance or the vapor-liquid equilibrium, but we did need some data for the heat transfer, for the heat losses, which indirectly, of course, will affect the energy balance and so on. And so you can see that we have some plant data, dynamic plant data, and which are the blue dots, and then the red dots are the model predictions. So you see we have a very good fit of the energy and the heat losses at the plant. The second was to configure the optimization problem. As I mentioned before, we are very flexible, so the operator can choose at the beginning of the batch which particular combination of operating costs, equipment costs, solvent costs that they want to use to make sure that it is optimal from the perspective of the business, and that may change, of course, from one day to the next. We, of course, have constraints. We cannot have too high a volume. We shouldn't have too low a volume. Jacket temperature needs to stay within certain ranges. And what we can manipulate is the rate at which we add the solvent, the new solvent. And also we can, of course, change the heat input by manipulating the temperature of the utility system. So the nonlinear MPC will make sure that all these variables are controlled, stay within certain ranges. We have a cost function that needs to be minimized, and we have the manipulated variables that have to stay between certain ranges, but we also have, we need to make sure that the model doesn't tell the control system to change flow rates very, very quickly. So you can see we have a maximum change in flow rate or temperature per 10 minutes. So this makes sure we don't get dynamics in the manipulated variables that are too fast. And then when we hook this up to the plant, because the main communication is here between the OPC server, which talks to within the DCS system. Here we have the actual model that was calibrated by the process development engineers. And then there is a piece of software which takes care of the plant data acquisition. It also deals with what to do if certain data is missing. If suddenly one of the sensors for temperature or something else doesn't work, it can make certain assumptions on extrapolating measurements or keeping them at the last measured value or some average of the last several minutes. So here we have some results of the one of the batches that was performed. Here we're looking at the reactor volume. So this was the first batch where the model was running online in parallel with the actual process. So the model is the digital twin because it runs in parallel with the actual. See, we had some deviations at the beginning, which was a result of foaming, which the sensor thinks that there is a significant increase in volume, but it's only foam. The prediction errors stayed within the validation standard. Similarly, for the temperature, we have you see very good correspondence, stayed within the acceptable ranges. The liquid composition, so this is something that is not measured, but the THF and propanol, the two solvents, you can see we are displacing THF, adding the propanol, and we have some water in here as well. and ethylene glycol. And this is very interesting because here you can see that the model predictions, which are the lines, were compared with measurements from samples. So you can see we have very good predictions of in real time of what is measured in the lab later. So this is of course done the first few times to make sure that the operators have confidence in the model and trust the control system. So you can see we have, you know, relatively very small errors, sometimes less than 1%, sometimes, okay, some of them 5%. So the benefits, when you compare the batch that was operated with nonlinear model predictive control, this was compared against the best batch that they had run for this product previously using manual optimization by the operators. So you can see we have a significant reduction in batch duration from 16 to 7 hours. We've reduced the amount of solvent significantly. There is also the reduction is higher than 28% because there is also a reduction in energy for this batch. So this is a very nice example because for a few reasons. One is because it uses standard thermodynamics. We need very, very little data to calibrate the model, really only to validate, and just using data from a plant trial. It's also interesting because, you know, if you want to try new techniques like nonlinear model predictive control, it's easier to try it on a solvent swap than trying it on the final API crystallization, because of course that is going to be much more critical for final API quality. So this is a good environment because in most cases, solvent swap does not have a significant effect on the final API quality. Okay, so that's the first case study. The second case study is is work that was presented by the FDA also at IFPAC, but not this year, but the year before, 2022. And this is an application of continuous direct compression. You can see there are 6 loss and weight feeders going to a horizontal blender. There is a surge hopper, a feed frame, which of course in reality is part of the tablet press. But when we model this, we model the feed frame separately from the actual tablet compaction. And the reason we do that is that there is a residence time distribution in the feed frame, in the surge hopper, as well as in the blender. And of course, the residence time distribution is very important for this system because the more residence time distribution, or the broader your residence time distribution, the more robust the process in terms of dealing with variability of API or other excipients coming from the loss in weight feeders. But it also allows us to better understand how all these fluctuations propagate through the system. And if we have too much residence time distribution, and we don't have a problem and we have to do a recall, then you may have to recall many, many lots. Yeah? So you don't want to have a residence time distribution that is too broad because that's not good for product traceability. If your residence time distribution is very narrow, then you have the problem that your process may be may suffer from robustness issues. Okay, so the objectives of the study that was presented by the FDA was to, first of all, develop and calibrate the flowsheet model that I showed on the previous slide. The second objective is to integrate the model. Yeah? So virtual plant is another word for the model or for digital twin. How to integrate it with the DCS system and the operator training system. And then, of course, they wanted to test the manufacturing process in a virtual environment using the DCS system and the operator training system. I already mentioned some of the details on the flowsheet model, so I will not repeat that. The HMI, the human machine interface, or the user interface for the operators, is in the DCS system. And so also 6 feeders in the blender and the surge hopper and so on, and the tablet press. But this is normally the interface to the equipment. And then we have the operator training layer, in this case, which we use to connect the flowsheet model, the virtual plant, to connect that to the DCS system. So that was done as part of the project for the FDA. And then once you have the connectivity, you can actually use this system in a virtual world, because they did this before they actually built before they installed their CDC line, to look at certain scenarios. So in this case, they were looking at having an increase in the compression force. And what you can see is that there is a response that you find the tablet thickness, of course, becomes a bit smaller. And the, what do we have here? The tablet hardness, of course, increases, which is the green line. So this is, I think, and this is important because normally operator training systems focus on filling, heating up, cooling down, and so on. This is an operator training system that actually predicts what the effect is of operating conditions on not process performance in terms of throughput, but also predicts quality attributes of the, in this case, of the tablet. You can also use this model to look at different feeder refill strategies. So here you're looking at a decrease in screw speed, which results in an increase here in the feed factor. And then the blue line shows you the refill of the feeder. And you can see how when you refill the feeder, you get some more fluctuations in the flow rate coming from the loss and weight feeder. And that brings us to the final slide. There are many, many more slides actually in the original presentation, but no time to go through them today. Also, the ability to use the interface in the DCS to control the mechanistic model to look at the effect of certain disturbances in flow rate from the feeder, and when the product should be diverted or when the product can be accepted. Okay, and then I'll just go to the next case study, and I'll try and wrap that up in 5-6 minutes, which is on biologics. This is a case study where we're looking at different sections, both the upstream, so the bioreactor, as well as the downstream, the chromatography sections. So we're looking here at a process where here we have the bioreactor. Initially, we operate this in a fed batch mode until we get to certain cell concentrations. And then we start running it in a perfusion mode. You can see when this harvest flow rate starts. And then because we are harvesting, we can also get even higher concentrations of cell concentrations in the bioreactor. After the upstream section, we then can model, and these models are all linked. So you'll see in a minute that when we look at disturbances in the bioreactor, the model will predict what happens also in the downstream section. So we're looking at the flow rate from the harvest tank. We have 4 chromatography columns, which are all operated in a periodic counter operated. And what you can see here are the traces of one of the columns that show, of course, the protein in A concentrations. And then we also can see what the actual purity and the recovery levels are that we get from from this part of the chromatography system. And then we have another chromatography system, the anion exchange chromatography. Again, we can look at the dynamics coming from the, so this is what we're getting from the tank, which receives from the previous chromatography section. And then we look at the UV trace, which of course gives us an idea of the concentrations of the proteins going through that final column. And then we can again look at the predictions of purity as well as the recovery. So with this model, it is used to look at how robust is the process to upstream disturbances. When do we expect that an upstream disturbance will cause problems downstream, and in some cases it may not. And of course, this information will allow us to divert material after the bioreactor or not. So we're first looking at a disturbance where there was an aggregation event in the bioreactor at midnight, Monday or Sunday actually. And then look at the aggregate levels and how they go through the system. Yeah, so we have the aggregation event happening in the bioreactor midnight Sunday, and then if we look at this harvest tank, you can see that there is a peak coming through about 12 hours later. We then also see an effect on the what happens in the chromatography columns and the Pro-A concentrations. And then if you look at the material in this tank, we can see that the purity of the product, the red line, starts to decrease over Monday night and Tuesday. But in this case, the final column, around the AEX, the anion exchange chromatography step, that purity remains steady. So this column is actually able to deal with the disturbances from the upstream section. So this shows good robustness of the process. The next disturbance is, we have a failure of the pump after the bioreactor, so for the perfusion system. We have a failure, for instance, on Friday night. It takes maybe one day to actually fix that. So we have a period where we are not withdrawing any material. So we have 1 day of no material being taken from the bioreactor. And we can see that that has an impact on the performance in the bioreactor. We get depletion of the nutrients because we're not, because we cannot feed more material, because otherwise the level would become too high. And you can see that there's a reduction in viable cells here, but it recovers by about Monday night. Yeah? So this is nearly 3 days later. But we do have some cell death and lysis. This all results in some additional aggregation. But again, if we look at the downstream section, we do have some effects of the disturbances here in the chromatography columns. But again, this AEX column is able to reject most of those disturbances. So this is a very interesting case study. There is a very interesting presentation by a company called Resilience in Boston, who have actually used the same approach to develop an end-to-end bioprocessing system. I wasn't able to show their results, but if you look in if you Google it, you'll find Resilience end-to-end bioprocessing. So this is a third case study, not one that we have in commercial manufacture. It's mainly being used to design and tech transfer the manufacturing process to manufacture, but not a real-time decision support system yet. Okay, so just to conclude, oh, that was a bit too quick, sorry. Right. My slides aren't responding the way I want. I'm trying to get to my conclusion slide. But anyway, I will read them out. So, so what I hope you've seen today is that when we talk about Pharma 4.0, digitalizing the pharma industry, that I have shown how mechanistic models, based on understanding of physics, chemistry, biology, can be used to digitalize the development of new drugs, of development of new manufacturing processes, also digitalize the tech transfer and digitalize the actual commercial manufacture through soft sensing, model predictive control, and so on. And the reason that this is an approach that more and more companies are using is that models are very efficient. It doesn't take a lot of time, so that's good for process development. We can do extrapolation. If we go from one piece of equipment to a piece of equipment that has different heat transfer area or different residence time, it is straightforward to update the model. We don't have to create lots of new data. So we require a lot less data. So in general, this is an approach that allows efficient transfer of knowledge that can be extrapolated from process development through tech transfer through to commercial manufacturing. And with that, I'd like to thank you for your attention, and I think we've left some time for questions. Thank you, and I can't see the chat, so I'm not sure if there are any questions.
That's all good, Sean. Yeah, thank you so much for that. I don't see any questions right now, so feel free to put one in the chat. One question I had for yourself was, do you see this technology being used much within the Singapore market, or I guess maybe just APAC in general?
Mm-hmm. Yeah, that's a good point. Thank you. So the answer is yes, it is being used, and I think especially thanks to PIPS, so the Pharmaceutical Innovation Program Singapore, that MSD, Pfizer, GSK, and also Syngenta are members of. We have seen a lot of use of the technology in some of the general PIPS projects, which are a bit more academic. But we have also done some projects that, for instance, Pfizer have done a project using this software for lot genealogy, so for continuous manufacture, taking into account all the residence time distributions and disturbances that you may have in a continuous drug substance manufacturing process. And also GSK have presented at several events on some of the work they have done to use these models for training operators, for soft sensing to inform the control strategy, and so on. So, if you have contacts within those organizations, or if you happen to work for one of those organizations, then there are definitely people who can talk about this from their experience as end users.
Cool. I don't see anyone typing, but anyone want to come off of mute and talk?
So if not, I will.
We got one question for you here, Sean.
Okay.
You shared an excellent EG on continuous manufacturing for OSD. Is it possible to have a model for a traditional tableting process?
Yes. So that's a really good question. So for all the and actually I have a slide here which may be useful. Not this one. I'll include these in the slide deck as well. This just shows some of the ecosystems we're involved in for synthetics and biologics. But if you look at the models, the mechanistic models that are available within the software environment, of course, we have a tableting model which can be used so nearly all these models can be used in batch or continuous operation. Of course, twin screw granulation is typically only continuous, but typically all the models shown here, if it makes sense in reality, you can use them for both batch manufacture and continuous manufacture. In some cases, you may have that, in some ways, tableting is a continuous process, but most of the other steps typically will be will be batch. Yeah? So we can model batch, for instance, high shear weight granulation and a batch fluid bed drying before doing maybe tableting, which you could consider continuous, and then a batch coating, tablet coating operation. So yeah, if you have particular applications in mind, be more than happy to answer an email or get on a call. And I think the other thing worth pointing out is that the technology that we're talking about here was originally developed by a company that was a spin out from Imperial College in London. I joined many years ago, 23 years ago, but since 4 years, that technology and myself are part of Siemens. So we also have actually colleagues in Singapore who also can help create connections and provide some initial support or knowledge transfer.
We have one more question here. It says, thanks for the insight. As stated, this is currently used for process design and engineering. Can this be used in the future for risk assessment or deviation management to evaluate the process risk downstream of a deviation step in any way?
That's an excellent question. And the answer is that some of the customers are starting to use these models for filings. So they don't put the model in the filing, but they will use some of the model predictions for design spaces to within the filing. Now, of course, some of the points of the design space they still have to validate experimentally on the actual equipment. But I expect that it will become increasingly more common, and especially because the FDA is driving this as well. Because when you look at, there are so many possible different disturbances that it is never going to be possible to actually test all the disturbances on the actual equipment. So models will be used to look at many more possible disturbances and whether the process is inherently robust, whether the control strategy is adequate to deal with lots of different failure or disturbance scenarios. And again, we had a user meeting last October in London, and there was a whole session on regulatory aspects ranging from using models for filing control strategies, but also looking at the GMP aspects, 21 CFR Part 11, for if you want to use the model in the commercial manufacturing. So the nonlinear MPC example I showed, that is now going into commercial manufacturing. Of course, the main thing that then has to be done is make sure that the GMP aspects are covered so that the right model is used, the right objective function is used, and everything is traceable. So yeah, thank you for that question.
That's about all we have time for, Sean, but do you want to leave us with any last thoughts or how to contact you, anything like that?
The last point is that I'll create a PDF of these slides, and I believe they will be made available through yourself, Jeff, or through ISP.
Yeah, we'll send it out to everyone.
Okay, excellent, thank you. And yes, the on the first slide is my email address. So anyone who's interested in contacting me should be able to get that email address as well.
Lovely. Really appreciate your time today, Sean. I think we all learned a lot. If you want to keep in touch with ISP, hit us up on our LinkedIn page. Check out ispesingapore.org for our conference, and look forward to seeing everyone again at our next event. Take care, everyone. Have a great evening.
Thanks, Jeff. Thanks, Sean. Bye.
Bye. Bye-bye.